The really neat thing about the enzyme that we put into this tree is it is one that people eat all the time. It's called oxalate oxidase, and it sounds kind of strange, but it actually comes from wheat. The neat thing about this enzyme is that it detoxifies an acid that the fungus that attacks the tree makes.
So today we're doing a oxalate oxidase assay, which will help determine whether or not a tree specifically is a transgenic tree, one of our transgenic trees. The purpose of this test is for anyone who is at home who wants to plant American chestnut trees and have them proliferate as opposed to die after 15 to 20 years, the ones that would not have the transgene. That's normally their lifespan. So you're going to need 6 tubes, 2 for this tree, 2 for this tree, and then 2 for your banana. What we're going to need to do first is collect our tissue samples. So I'm going to pass out a bunch of bananas. If you just put a little piece in both of your B tubes, so B plus and B minus, and try to do a little bit of an equivalent size if you can. Yeah, that's perfect. So a piece of that size in each one of your B minus and B plus tubes. There we go. So, nope, it doesn't matter what part of the leaf you take, you just want to avoid the mid vein. So it's just going to want to be something on one of these sides here. So I'm passing out leaves right now that are the pink labels, so don't mislabel them, otherwise it'll seem like I'm wrong. Well, you don't even have to.
If you take the top, and push it down, and then it really go through.
So there's a negative control and a positive. So say you got a black ring in both your negative and positive solution for one of these trees, that would mean that there's some sort of contamination, maybe some plus solution got into the minus. In that case, it would be a null and void experiment and it would prove nothing. The theory behind this is that there are a negative control and a positive control, and if our tree has oxalate oxidase is the gene, and that codes for oxalate oxidase, the enzyme, and that is what breaks down the toxic agent of the fungus, which is oxalic acid. And the oxalate oxidase breaks down oxalic acid into hydrogen peroxide and CO2. And both of those are beneficial for plants when they're wounded because the hydrogen peroxide helps with cell wall lignification, so it basically walls off the cell and protects it from the acid stronger than if it were just a regular cell. And then the CO2 is just a natural byproduct of the plant anyway, so it's not a big deal. It's not the best thing to have on skin contact. When you're done, you have a bag like this. You just want to shake them up and make sure that they're all in the solution. And then just put them in your bag or wherever you want to store them that's out of light. And then we'll check them at dinner. And then we'll see our result. You can just take these 2 little hole punches, put one into a positive, one into a negative solution. In the positive solution, you would have a blackened ring formed by the hydrogen peroxide that's being produced as a byproduct of the oxalate oxidase enzyme breaking down oxalic acid, which is found in both solutions. And so the purpose of that is, so when you grow up 100 trees, you can choose the best ones, the ones that will be resistant, to plant in the nice areas, to plant wherever you want, and then you can use the ones that aren't transgenic as mother trees to produce more seedlings that are transgenic and are not. Because of inheritance, only 50% of your offspring are going to be transgenic and therefore resistant to the blight as opposed to the mother trees. We're producing the transgenic trees, but for them to be restored in the natural environment and for everyone who wants them, we can't produce all those trees that's needed. We're going to need seed orchards, we're going to need people who are willing to plant these trees and collect nuts. So that is the reasoning behind this quick and dirty test, so people don't have to send leaves to us by the hundreds from their orchard. They can just go out, put little discs in their tubes, and see.